You're seeing the screen?
Yes, sir.
Okay, good.
Yes, sir.
Yeah, thanks. So in the last class, we have learned about, in the last classes, we learned about the properties of embryonic stem cells. Then we came to their derivation, the technical parts. And then we have discussed about a little bit about applications and so on. In derivation, we learned about IVF and then the techniques of establishing an environmental, in embryonic stem cell technology. And briefly, we touched upon applications and we came to what are the problems associated. The problems. So basically, it is if we are learning something and we know we are trying to find or enhance the embryonic stem cell technology, then it's not just that we know the greatest things or the positive things of it. We should also know about the problems associated with it. If you think of any knowledge, it's something like this. If we don't know what all we know, or I should rephrase it, sorry. If we know what all we don't know, then we know everything almost, right? So then we will certainly have a lot more to know, and we will keep on learning. That's how we remain learners for life. So as a part of trying to know the problems or gaps in knowledge and technology, then we have come across about for embryonic stem cell technology that there are problems with immunorejection, there are problems with undifferentiated cells, and so on. Immunorejection, we had solutions such as these, that is somatic cell nuclear transfer. That is, we are solving this problem by taking patient nucleus and developing embryonic stem cells and deriving whatever cell type we want and then putting into a patient. So these are the cells will have absolutely identical genotype as that of the patient, and therefore there will not be any immunorejection. That is one of the points of it. We also learned about the problems with rogue undifferentiated tissues, right? Or the embryonic stem cells, which may actually cause teratomas like this. Anyway, continuing with the CNT, we saw that stem cell technology, SCNT is powerful, but at the same time it has its own technical and biological issues. First one, it is very inefficient. Then we will learn about what it means to be epigenetic programming. We will learn about it, so I will not touch upon it right now because it's a whole huge topic to start with, and we have to start from fundamentals, so we'll leave it there. And the number of eggs are insufficient, so people suggested animal eggs, but then there are problems with belief systems, ethical issues, and so many other stuff. And in that, we also learned about the disharmony between the nuclear mitochondrial genes and the mitochondrial genes themselves. And there are problems with aneuploidy or broken chromosomes and so on. We learned about G0 as theoretically, at least, is a good or a stage for employing in SCNT because it is in 2N and everything, the genome is probably the best possible way. So the alternate approaches to prevent rejection, right, apart from SCNT, because there are problems with it. We are damaging some embryos. We are in the between making a whole animal, human itself, like the reproductive cloning we have discussed. This is the other path. If the blastocyst is implanted into a surrogate mother, it can give rise to an organism that is the clone of this patient. That will lead to social and legal problems. So the other ideas provided were, say, if we had the graft rejection typically happens because of MHC molecules and so on. And if we try to see what are all the combinations possible for MHC and so on that are involved in immunogenicity, there are about 450 combinations. What if we could all collect different embryonic stem cell lines from all over the world and generate a repository or a stem cell bank, human embryonic stem cell bank, such that all combinations of MHCs are present? Then if there is a patient that comes by and wants a certain matching cell line, probably one of it is likely to be um, a good match for the patient here. So that is one approach, and it's a very viable approach as long as we get the diverse haplotypes. Then there is another approach is people suggest that, say, this MHC, this is the gene, and if we can make some genetic manipulations such that the MHC on the surface does not arouse any immunogenicity when implanted into a patient, right? To try to make it a universal donor kind of human embryonic stem cell lines. So that's another approach, but this is just it's an idea. It is a good idea, but it's impractical for a lot of reasons. The other one is some people suggest that, okay, if you have problems with it, you take the embryonic stem cells, encapsulate them in membranes that are immunoprivileged, meaning that they will not provide access to immunocomponents such as antibodies or maybe the macrophages and other things. Thereby, the cells that are implanted will remain protected from the host immune components. This is also an idea, but I don't think it is. I don't think it. In my opinion, it's not the best of the ideas, because the cells need nutrition, and there should be supply of blood, and there should be many things that are signals and so on that should go into. It's not that it won't go, but to make these the connections with the rest of the body, we would, anytime we implant some cells into the body, we hope that it will get integrated into the body, become as part of the body and functions in coordination with the rest of the tissues. If we are encapsulating, maybe it's not so integrated. So that's why, nevertheless, it is an idea, and it's worth knowing that it is a possibility at least. And then we can probably use similar ideas about, we have a problem, and this is a brainstorming session where we try to find out what are all the possible ideas. If it were in a theoretical, in physically present in a classroom, I usually also has brainstorming as an exercise. Maybe if we meet together, we will have that session someday. So the next problem we have until now we have tried to sort out the problem or address the problem of immunocompatibility. And now we are trying to sort the problem of undifferentiated cells. The problem being that we have embryonic stem cells, we derive the cells or differentiate cells of our interest, and we would want to give to the patient. The problem is, if we have taken 100 cells, maybe 95 have differentiated, but 10 of them or so, or 5 of them may have remained as embryonic stem cells. If these also are implanted, they might give rise to a teratoma, and that teratoma could kill the person. So we should be able to somehow get these out so that we only give the differentiated cells, but otherwise not. So the focus of today's class will be about FACS and MACS. Okay, we will see in the next slides, but I would want to also touch upon the other ideas that are present here. One of the idea is to put in apoptosis controlling genes or so. So I am sure, yeah, I think you haven't studied genetic engineering of such sorts. So, but molecular biology wise, you might be able to understand a little bit of it. See what is possible. Assume that this is a regulator of apoptosis. Say the protein would somehow result in apoptosis. Apoptosis is kind of induced cell death. I mean, suicide-like thing. And if we have engineered a promoter, this is the promoter, right? And that promoter, you're putting it, you have chosen a promoter that will be open to regulation. It is, or else it is inducible only in embryonic stem cells. What would have happened to it if the cell has undergone differentiation? You would have chosen the promoter that I'm talking about, such a way that in the differentiated cells, this promoter would become silenced. Okay. Now, say for example, if you have already made such a genetic manipulation and put it into embryonic stem cells, and you have a mixture of cells. Say for example, you have embryonic stem cells and you have a majority of them differentiated cells. Now I said this promoter is inducible. Say for example, I'm going to add XYZ, and after differentiation you have a mixture of cells, right? And I have added XYZ. What would happen? In differentiated cell, the manipulated gene I was talking about is silenced. So the XYZ, even if it is present, it will not get activated. So differentiated cells will survive. Whereas embryonic stem cells, the promoter is active, and if it is induced by XYZ, it will result in the formation of a protein that induces apoptosis, and the embryonic stem cells would be killed. So now we can take these differentiated cells and use it for therapy. That is the concept of what is given here, like this. The basic concept, okay? Or else the other solutions are to try to encapsulate all these cells into with alginate beads like thing. It's something similar to that of previous one that we have said. So hopefully the cells, if there are any embryonic stem cells, they can be they might not escape in the form a tumor into the other bodies, or maybe the capsule is going to provide some hindrance against excessive growth. But that's not, I don't think it is a great idea, but it is still an idea. So there is another way of trying to select, that is using selection using antibodies. Just I'll erase some so that we will be able to understand what I wanted to say. So in this case, what we are going to do is, you know that if you're reading immunology now already, or cell biology, you know that the cells have cell surface markers. What are they? They are just some receptors or some proteins that are on the surface of cells. So say for example, embryonic stem cell has a receptor called A, or some protein that is available on the cell surface. And if it differentiate into some other cell type, okay, these two are different cells, right? They have the same genome, but their gene expression is different, because of which their transcriptome and proteome will be different. So maybe the differentiated cell will have a different receptor on surface of it. Now, what the suggestion in this selection is that if we have made antibodies against, the antibodies look something like this. You could have made an antibody that is cytotoxic, conjugated with a toxin. Okay. And this antibody is anti-A antibody. That means this is the anti-A here. This is the A antigen. So if you add a mixture of this antibodies to a mixture of cells containing embryonic stem cells and differentiated cells, then if the antibody is added, it will bind only to the embryonic stem cells because we have chosen a receptor that is present in embryonic stem cells but not in the differentiated cells. And because we have conjugated this antibody with a toxin compound or a molecule, it will kill the embryonic stem cells, and then we are left with the differentiated cells which can be used for therapy. So these are the kinds of approaches. In one of the lessons we learned a long time ago during the first classes of molecular genetics, Hershey and Chase experiment, I guess, where we have used density gradient ultracentrifugation. Or the centrifugation is basically, where there we have discussed something like this. You can use analytical centrifugation or preparative, you can call it, where you would have made a density gradient in the centrifuge tube. So high density here and low density here. Okay. And if you layer the cells, a mixture of cells, I hope you can also recollect that the embryonic stem cells have high nucleoplasm to cytoplasm ratio, whereas the differentiated cells are much more, have something like this, right? They certainly have differences in their shape and compactness, and therefore they will have different densities or buoyancies, which when the mixture of cells is applied here and we perform centrifugation, maybe the embryonic stem cells will come here. It depends, I'm unsure. Embryonic stem cells, say for example, may come here and differentiated cells may come here. It depends. So this technique is trying to capitalize on using the differences in shape, buoyancy, or density, and all those things. So now once we have performed that, we would get differentiated layers cells in one case and the embryonic stem cells in one case. Now we can collect from here, we can collect the differentiated cells and use it for therapy. So that is another solution to the problem, which is a serious problem, right? It might otherwise give rise to teratoma. So what I will do is right now is focus on to the cell sorting. This technique is amazing. Since the time I have learned it, it has been very fascinating to learn about it. To appreciate bioengineering to start with, or biomedical engineering, and how much technology, the ideas people have, and which we otherwise think it's not possible, these people make it possible. This is called as FACS. CS is for cell sorting, right? Fluorescence associated, I think. Different people have different ways. Some people say it as activated, some people say it as associated. So this technique is about cell sorting. What do you mean by cell sorting? Say, for example, you have squares and these like this. This is a mixture of things, and you want to sort them into two different boxes. And I hope you know it, right? But let me just take it. If you are able to sort all these squares and circles like this, that is sorting, right? The same thing we want to do. Say if we have a mixture of cells and we want all the embryonic stem cells into one place and all the rest of the cells or the differentiated into another tube, that is sorting. It is cell sorting. And cell sorting can be done in many ways, two of which are, one is fluorescence associated, the other one is magnet activated cell sorting, right? So the principle is in the term itself. So otherwise they are called as FACS, FACS. So what we need to do, like we have discussed in the previous one, is we will will use different antibodies and cell surface markers. So we need to go back, like what we have discussed for antibodies, the same technique can come in handy here. So I would choose, this is the embryonic stem cell, and here would be this is, say, for example, differentiated cell. I may have a antigen A, and on this, it may have a receptor or a cell surface protein called B. So I would raise antibodies against it. How would they do it? They usually put it into a horse, or they collect some proteins and they inject into the horse blood, or many guinea pigs and so many other things. Other animals also could be used. So the horse will raise antibodies against the protein. Say, for example, I used protein A, right, that is present in the embryonic stem cell. And then they collect the serum of the horse. From there, they would have isolated antibodies against A. So you call them as anti-A antibodies, like that. If you want to generate antibodies against B, you would have injected antigen B protein into the horse. So in about some days, the horse will generate antibodies against B. They would be being called as anti-B antibodies. Okay. So there is something else that we do for antibodies here. Like I described about the antibody being tagged with the toxin in the previous topic. So here, what we do to these antibodies is that we tag or conjugate a green fluorescent dye, right? If we put it into laser, this would give out green fluorescence. For antigen B, we would have tagged with red fluorescent dye. So basically, the antigen, if we so let me see this. Let me try. So here you would have had green or antigen or red. So this is anti-B, and the top one is anti-A antibody. So if we add this to the types of cells that we have, a mixture of cells, A will get tagged with the antibody which is having red fluorescence. It's a green fluorescence dye. And B would be, or the differentiated cell would be tagged with antibody, and that is containing the red fluorescent dye. That is the sample preparation. What we should also to ensure it is that it is a single cell suspension. That means there are no clumps of cells. They should be free, individual, independent cells. Second thing is we should have maintained their concentration in a way such that they would remain in single cell suspension, and there are other considerations as well. In today's class, we will only generally try to understand the basic principle. Over the period of time, you would understand it more and better. Okay, so don't pressure yourselves to understanding everything right away. It's all right if there was a lacuna anywhere. So that is the preparation one would require for making the cells, and these cells will be added into the machine. The suspension, the machine, the tube, the container has a very narrow, fine tube through which at any given time only one cell will pass through, right? And there is a incident light of with the laser. Laser is being shot at continuously, and every time a cell passes, based on because of the laser, And because of the antibody, the color fluorescent tags that are attached, they would give either red or green. If there is red fluorescence, that means it is the differentiated cell. If it were giving green fluorescence, that means it is embryonic stem cell. It doesn't matter to the computer. We would only, the detector will only see what color is it, red or green. And if it has received a signal and the signal is sent to the computer, computer is programmed to say, if it is red color, if it is red, give negative charge. If it is green, give positive charge. That means when the cell passes from here till here and it forms a small droplet here, a charge is added to that drop, right? If it is green, it will be given positive charge. If it is red, it will be given negative charge. So one droplet at any time is having some charge. If there is no fluorescence at all, then there is no charge is given. Always we should remember that we are adding the antibodies. It's not sure. There is no guarantee that all the cells will get tagged. Some may get. Sometimes there may be a scarcity of antibodies. There are not enough of antibodies to bind all the cells, or there could be some are just they didn't mix up well. So as the droplets pass through this way, if they are negatively charged, they would get attracted towards the electrodes that are placed here, towards the positive electrode. And so the path of the droplet should have been like this, but because of this deflection, they would change the course of path and they would fall in this tube here. If it were a green cell, it would have gotten positive charge, because of which if there is green fluorescence, because of the positive charge, it will deflect towards the negative electrode and is likely to fall in this one. If any of the droplet is not, there is no fluorescence, then the computer will not give it any charge, so it will fall straight down. So I hope you remember what I started with. We have a mixture of cells, and we should be able to sort them into separate containers. And this is exactly what is happening in this case. Okay, it is a fantastic technique. We will watch some videos today, and probably that will benefit or better your understanding. This is called fluorescence associated cell sorting. The other one is called as MACS, or magnetic activated cell sorting. This is a much simpler and easier one. Here we would do embryonic stem cells, and we have differentiated cells. So we take antibodies against any of them, and have antibodies are engineered such that are conjugated with magnetic beads. Okay, and when we add, say, this antibody will have only specificity, for example, against only the differentiated cells, right? Or you can also choose to have only specific to embryonic stem cells. So for right now, I will just go with that. Assume that these are anti-embryonic stem cell type antibodies. Okay. So we made the mixture of cells. There should be a single cell suspension. We don't want clumps to be there or aggregations. And this is the column. It's a column. It's something like a tube. And we add the cells through it, which has a gel pack and all. So the cells will are very little bit to hold on. And these are the magnets that are present, right? So the cells that are poured will keep on moving down. They would stop, however, if any of the cells have the magnetic beads. The magnets or the beads will be retained in this position here, because they are conjugated to antibody, and if the antibody is there, the cell to which they were raised against to, say for example, embryonic stem cells, ESCs will remain here and the rest of the cells will flow through, and you can probably use them, right? If you later, if you remove the column from the magnets, these are the magnets, north and south pole, say, and if you remove them, and then the rest of the cells will also come here. You have done sorting. One type of cell is here and the other type of cell is in a different tube. Now I have a question, is which cells to label? Say the objective is, if a researcher wants a pure sample of differentiated cells, which cells should he or she label? Is it better to label embryonic stem cells or differentiated cells? That's the question. Anybody wants to try?
It should be differentiated cells, so that only the pure sample will be obtained. Because if we choose the other one, there will be a mixture of differentiated cells, and some receptors which are not bound to may also enter the sample we are considering.
That's exactly right. You're right. Your name, I can't see yours.
Rishis.
Rishi, okay. Thank you. So when we add these antibodies, anti-ESE embryonic stem cell, for example, there is no guarantee that all the embryonic stem cells will be labeled or bound by the antibody. What may happen in this case, like I already described, say we have labeled embryonic stem cells, and now we have collected apparently differentiated cells, but there is also a risk of unlabeled embryonic stem cells or unbound embryonic stem cells. And if you give this to a patient, there will be a problem. Instead, if we label the differentiated cells, so the differentiated cells would be retained because of the magnets, and the rest will flow through into a tube. What may be there? Embryonic stem cells and unlabeled differentiated cells. Losing differentiated cells is not a problem. We want to get rid of embryonic stem cells. That is the goal, because even one of it would jeopardize the whole concept of embryonic stem cell technology. So if we have labeled the differentiated cells, and then we would get rid of embryonic stem cells, then we can take the column out of the magnets, and then we could probably get only the differentiated cells. So I'm not trying to imply that this is the way that it should be done. What I'm trying to communicate here is that we have problems. This is a problem solving skill or troubleshooting or trying to generate the best solution. So this is what we want. I want a pure sample. Excuse me. Sorry. So I want a pure sample of differentiated cells. That means I don't want even one embryonic stem cell in it. We cannot guarantee it in practically, but theoretically, this is the best solution for the problem that we have. So when we try to solve a problem or come up with an idea, the point that we should see is what are the requirements or constraints that are given are there in the problem. Then if we know them and we acknowledge them, then we can find the best solution with the available resources. Okay. So I'll stop here, and we will watch some videos, and then probably we will see. Sorry. So I'll go with this first. So I think you cannot see the animations, audio. The flow cytometry has something like a mesh. It's just a big box. Let me see if we can go. You can use it for many purposes. I am only using it here for our purpose. It's just 1 or 2 sentences that are mentioned in our textbook. But it's worth knowing it because it's an amazing technique as well. So this is how the the fluid keeps coming in here, and this is where the laser is, and this is the detector. Please remember that there are multiple points, or it's a complex technique. It can be used for preparation, or it can be used for analytical one. We are only discussing about preparative one. For analysis, you have to learn about forward scatter, side scatter, and many other things. But right now, we will just focus on that. I mean, this technique might recur in some more chapters. There we will discuss. Gradually, we'll keep increasing the complexity of what we understand about flow cytometry. So this is how a droplet keeps falling, and then the laser will keep doing that. And when there may be side scatter or forward scatter, and for our purpose in that, we are only using it for fluorescence, right? The machine can come with different types of detectors, and each detector, one second. So it can, if you have just the forward scatter concept, you can also have only for forward scatter. It depends. We will discuss about it later, maybe if required. But otherwise, we can also detect colors and so on, like for fluorescence. So this is how probably the cells will look like, and we have to. Why I'm showing you these is because you are bioengineers. And if you want to construct anything, then you have to take into, say, this one picture has so many things. It's about the shape of the device. It's about the flow of the fluids. It's about how the cells are maintained as single cell suspension and they move through unhindered and so on. I hope, yeah, we'll leave this video here. Where is the other one? This is how, yeah. I will send also the link on the WhatsApp group. You can check. So this is how a flow cytometry graph would look like if you are measuring the different types of cells. Single cell suspension is taken, and it will be put into this place where through suction, the whole sample is taken up into the machine. And then the cell suspension will go through one cell at a time. If the laser or the light will hit, and based on what you are setting up, either the scattering or the color, you can choose whichever one you would like it. And then they can be, the detectors will send to the computer, and the computer will do the rest of times. Sometimes we just want to know the types of cells, heterogeneity within the sample. Sometimes we want to sort. Sorting is an additional thing. This happens very fast. In 2-3 minutes, the whole thing will be done almost. So, but yeah, one cell at a time. This is forward scatter. Whatever is scattering here is side scatter. This is side scatter. If you have a detector here, that is, it is detecting only the forward scatter, right? The cell size, different sizes will have different scattering patterns. Based on that, it can detect whether it is cell is big, small, and so on. So each time there is the signature, the detection will be sent to the computer, and the computer will keep mapping or trying to show the cells. Say, for example, this is the side scatter detection. Side scatter may help for the with the shape and internal components and other things. If you watch this video, you probably will understand it better. I'll send the link. Yeah. Sorry. The other one is magnetic cell sorting. Um, this is simple. As I said, you have the magnets here, south or north pole, and then the sample is injected into this column, and then magnet is added, and magnet is present anyway. So the cells that are with here will be retained, and the rest will flow through. So they can be used for multiple purposes. And MACS is very easy and better because it is simply portable. You can just take it and try to use it anywhere you want. Whereas MACS is an expensive machine and you need a trained, very well trained person to handle and operate the whole machine. MACS is simple. You would, I think Shastra has FACS. I hope you will, as a project student or so, you may get to use it. Any questions in this? In the next class, we'll summarize and we have some topics to discuss, and then we'll conclude it. If you have any questions, you are free to ask.
Is there a technique that we can perform after diagnosing a tumor that is caused by?
That is caused by? Completely. I didn't get the question.
Just a sec. Sir.
Yes.
So, which is caused by the undifferentiated cells, like after diagnosing it.
No, yeah, certainly. The point is that not if you take any tumor, okay, there will be say 4 or 5 types of cells, all belonging to the same lineage. Right? If it is from, if the primary tumor, you say, I mean, it will get complex, but let me try answering. Say if a tumor has arisen in one body part, say skin, okay, a melanoma or so, and it has metastasized and reached several other organs, including the lungs, say, for example. So this is where it originated is the primary tumor. These are the secondary tumors and so on. They have arisen from the primary tumor, and therefore it will have different, at least say 3 or 4 cell types, okay, all belonging to the epidermal type. Okay, even the secondary tumor will have characteristics of the epidermal one. That is how they figure out where did this tumor arise from. So that is how a general tumor is. And teratoma is different because it has the cell lineages derived from ectoderm, mesoderm, and endoderm, all the 3. It's almost like a disorganized or unorganized fetus growing in the body. And that is one of the reasons why they also say developmental totipotency to this. Okay? So because they have the potential to give rise to almost like a human body within a human body. So these tumors have arisen from germplasm. That is something like embryonic stem cells also have that potential, right? Pluripotent. Whereas other tumors do not have this. That is how we can figure out if these if these tumors have arisen from embryonic stem cells or not. We'll also have several in other lessons also how to characterize something is an embryonic stem cell or not. Anyway, you asked. I hope did I answer the question?
Yes, but
Yes.
I wanted to know if these mentioned techniques could be useful
Oh,
for
okay. For what?
after diagnosis of a tumor, say cancer even, because that's 2 immature cells continuously dividing, right? So
No.
can't we
What do you
use?
want to do after, say, you have the tumor? There is a tumor,
Yes.
and you do cell sorting. And what do you want to do here after that?
So once we sort, we will not have a tumor anymore, right? Because we
No,
have
where did you
filtered.
get this sample from? The tumor is already present in the body, you're saying. Is that right?
Yes, sir.
Tumor is already in the body. You had to cut it out. Is that right?
No, no. I'm talking like if it's,
Uh,
you have to purify your blood from the cells that are causing the cancer.
Okay. But
So
how will we remove the complete? How
yeah,
can it be complete?
but if it's not, I don't know if that's the term malignant, I guess.
Yes.
Yeah.
I mean,
So
malignant means it has moved from its primary site to different other sites.
okay,
It
so
has mixed up with the blood.
opposite to malignant. I don't know the term
No,
exactly.
that's all right. That's all right. Don't worry about.
If it's stationary, we could use one of these techniques to purify the blood from that part alone, so that it will not transport to somewhere else. And
Yes, that is possible theoretically. But how can we ensure that, say, for example, in all of blood, we have 6 liters or so of blood? Oh, sorry, 6 liters of blood. Okay. And how can we ensure that the tumor cells that are present in it could be, say, 100 of them? We can never be sure that we are able to take it out. First thing is that is the problem. Second thing is, if you are doing cell sorting and then you take the remaining blood, you have to give it to the person. Is that right?
yes, sir.
You should inject back into that.
Yeah.
Yeah, that is a huge problem with contamination and other things.
But we do a similar
Yes,
thing for
for kidney.
dialysis and
That's right.
other
For
stuff.
dialysis, we do it. We make it as one single thing. There is an inlet, and there is an outlet of blood, and there is an inlet machine, and nothing else is there in between. Is that right for dialysis?
Yes, sir.
For this one, who will make the single cell suspension? And blood is very complex. It has many other stuff also. What we put here is very important. It should be a pure sample of the cells. It cannot have other types of proteins or red blood cells, platelets. You have too many stuff in there. So if you want to do, I'm just taking it up because we can have a discussion on that. So if it is blood, you should have purified the cell components from the rest. Otherwise, they're going to interfere. And if you want to make a cell suspension, single cell suspension of for the sample in cell sorting, you should have diluted it at least 1,000 times. Okay.
Yes, sir. Got it.
So that means there is a human interference here. And then if you have, say, 160, that will become about 600 liters or so. That's on the lesser side. And after cell sorting, you should have concentrated it back into 6 liters, right? And then given to the person. So the chances of contamination, this is just for an idea, and to discuss about this, that is when we will know what are the constraints, and that is how ideas come up. It is a good idea to start with, to discuss on. Okay. Did you understand
Yes,
what I
sir.
said?
Got it.
Yes. Any other questions? Okay, then if no questions, then we'll stop here. Okay, take care and have fun.
Thank you, sir.
Thank you, sir.
Yes.
Thank you,
Bye-bye.
sir.
Bye.
Thank you, sir.
Bye.
Thank you.